Long before dinosaurs claimed their throne, before mammals rose to dominance, before birds painted the skies, there was an age so alien, so impossibly strange, that it feels more like science fiction than Earth's own history. Picture a world where dragonflies the size of seagulls patrolled ancient skies, where millipedes grew longer than crocodiles, and where scorpions hunted with the mass of large dogs. This wasn't some distant alien planet. This was Earth during the Carboniferous Period, an epoch when insects didn't just exist, they ruled. The air buzzed with wings spanning 2 feet across. The forest floors crawled with armoured arthropods that would dwarf anything alive today. Evolution had taken the basic insect blueprint and pushed it to extremes that defy modern understanding. These weren't just bigger versions of today's bugs. They were apex predators, ecosystem engineers, and evolutionary experiments that would never be repeated. If you've ever wondered what happens when nature gives insects the keys to the kingdom, prepare yourself for a journey into the most bizarre chapter of life on Earth, the age of insects. The oxygen revolution that changed everything. The stage for the age of insects was set by a chemical transformation so profound that it rewrote the rules of what life could become. Around 360 million years ago, Earth's atmosphere began a dramatic shift that would enable creatures to grow to sizes previously impossible. The Carboniferous Period earned its name from the massive coal deposits it left behind, remnants of vast swamp forests that covered much of the planet's land masses. These forests were unlike anything before them. Towering tree ferns, scale trees reaching over 100 feet tall, and dense undergrowth that trapped sunlight in a perpetual twilight at ground level. What made these forests revolutionary wasn't just their size, but what they did to the air itself. Plants had recently evolved lignin, a complex organic polymer that made wood rigid and resistant to decay. This innovation allowed trees to grow taller than ever before, but it created a problem for the ecosystem. Almost nothing could break lignin down. When these giant trees died, they didn't rot. They fell and accumulated in massive layers across swampy lowlands, locking away the carbon they'd pulled from the atmosphere during photosynthesis. This carbon sequestration happened on a scale never seen before or since. The consequence was extraordinary. As carbon dioxide levels dropped, oxygen levels soared. Modern Earth has an atmospheric oxygen content of roughly 21%. During the peak of the Carboniferous, oxygen levels reached 35%, nearly 2/3 higher than today. This oxygen-rich atmosphere fundamentally changed what was physiologically possible for air-breathing animals. Insects, which breathe through a network of tubes called tracheae rather than lungs, had always been limited by how efficiently oxygen could diffuse through their bodies. Higher oxygen concentrations meant those tubes could supply much larger bodies. The size ceiling lifted, but the oxygen revolution did more than just enable growth. It transformed entire ecosystems. Wildfires became more frequent and intense in the oxygen-rich air, creating a landscape of constant renewal where new growth exploded from burned areas. These fires may have been so common that some plants evolved fire-resistant adaptations, much like modern eucalyptus or pine species. The combination of abundant plant life, warm global temperatures, and high oxygen created perfect conditions for arthropods to diversify and dominate. The increase in oxygen didn't happen overnight. It was a gradual process spanning millions of years, driven by the relentless photosynthesis of Carboniferous forests. As oxygen accumulated, organisms adapted to exploit it. The first giant insects appeared as oxygen levels crossed critical thresholds. Early dragonfly relatives began to grow. Millipedes lengthened. Scorpions bulked up. Each generation pushed slightly larger. Each millimeter of growth made possible by another increment of atmospheric oxygen. Scientists discovered the connection between Carboniferous oxygen levels and giant arthropods through careful study of fossil insect anatomy and atmospheric modelling. By examining the ratio of carbon isotopes in ancient plant matter and marine sediments, researchers reconstructed past atmospheric compositions. When they compared these oxygen curves to the fossil record of insect sizes, The correlation was unmistakable. Giant insects appeared precisely when oxygen levels spiked, and they vanished when those levels dropped again in later periods. The mechanism behind this size increase involves the physics of gas diffusion. Insect tracheae are passive tubes. Insects don't actively pump air through them like mammals pump air through lungs. Oxygen simply diffuses from the outside environment through progressively smaller tubes until it reaches individual cells. In low oxygen environments, this diffusion can't penetrate far into large bodies. But when oxygen concentration increases, diffusion becomes more efficient, allowing the system to supply tissues much deeper inside the body. Mathematical models show that at 35% oxygen, tracheal systems could support insects roughly twice the linear dimensions possible today. This oxygen abundance also affected insect metabolism. With more oxygen available, insects could sustain higher metabolic rates, powering larger muscles and more active lifestyles. Flying became easier because flight muscles could work harder without running out of oxygen. Predatory insects could pursue prey with greater endurance. The entire pace of insect life accelerated, creating an arms race where size, speed, and efficiency all increased together. The swamp forests that drove this oxygen revolution covered vast territories. In what is now North America, Europe, and parts of Asia, these wetlands stretched for thousands of miles. The dominant trees weren't anything like modern forests. Lepidodendron or scale trees grew to massive heights but had shallow root systems, making them unstable in storms. When they fell, they created massive logjams in the swamps. Calamites, ancient relatives of horsetails, formed dense thickets along waterways. Seed ferns with massive fronds filtered the intense tropical sunlight. Beneath this canopy, the forest floor was a maze of rotting wood, standing water, and thick leaf litter. This environment was perfect for arthropods, humid, warm, and filled with decaying plant matter that smaller insects and millipedes could consume. The abundance of detritivores supported populations of predators, every ecological niche filled with specialized forms. Some insects evolved to bore into the tough bark of scale trees. Others developed aquatic larval stages in the endless swamps. Still others took to the air, where competition was less intense and new opportunities awaited. The oxygen levels peaked around 300 million years ago, then began a slow decline as ecosystems changed. Fungi and bacteria eventually evolved the ability to break down lignin, causing dead wood to decay rather than accumulate. This shifted the carbon cycle back toward releasing carbon dioxide into the atmosphere. As oxygen levels dropped, the giant insects began to shrink. By the end of the Permian period, atmospheric oxygen had fallen significantly, and the age of giant arthropods was ending. But for roughly 60 million years, insects lived in a world custom-made for growing large, and they seized that opportunity with spectacular results. Meganeura, the 2-foot dragonfly. Imagine standing in a carboniferous swamp at dusk as shadows lengthen between the towering scale trees. The air is thick and humid, filled with the buzz of countless insects. Then you hear it, a deeper sound, a rhythmic thrumming that vibrates through the air like a helicopter rotor. You look up just as Meganeura sweeps overhead, its wings spanning over 2 feet, each transparent membrane catching the fading light. This wasn't a dragonfly in the modern sense. It was an apex aerial predator, the undisputed master of carboniferous skies. Meganuropsis represents the largest flying insect ever discovered, with wingspans reaching 70 centimeters in the largest specimens. The fossils were first unearthed in France in the late 1800s, and when scientists initially studied them, they struggled to believe what they were seeing. The wings alone were massive, preserved in exquisite detail in fine-grained sedimentary rocks. The venation patterns, the network of supporting structures in the wings, showed remarkable complexity, with thousands of tiny cells forming a strong yet flexible framework. Unlike modern dragonflies, which are already impressive flyers, Meganeura was built on a much grander scale. Its body was thick and powerful, with a robust thorax packed with flight muscles. The head was dominated by enormous compound eyes that wrapped around the sides, providing nearly 360-degree vision. These eyes likely contained thousands of individual lenses, giving Meganeura the ability to track fast-moving prey with precision. The mouthparts were equipped with powerful mandibles capable of crushing other insects, and gut contents from related species show they fed on a variety of smaller flying insects. The wings themselves are marvels of natural engineering. Each wing was supported by a complex network of veins that distributed stress evenly across the membrane. The leading edge of the wing was reinforced with thicker veins, providing rigidity during the downstroke. The trailing edge remained more flexible, allowing it to bend slightly and create efficient thrust. This combination of rigidity and flexibility made Meganeura an agile flyer capable of rapid turns, acceleration, and hovering, all crucial abilities for a predator hunting other flying insects. Flight mechanics studies using computer models reveal that Meganeura likely flew with a slower wing beat frequency than modern dragonflies, but each stroke displaced more air due to the larger wing area. This made it energy efficient for cruising, but still capable of bursts of speed when attacking prey. Wind tunnel tests with scaled models suggest Meganeura could reach speeds of perhaps 40 km per hour in level flight, with even faster diving attacks possible. For an insect, these are extraordinary speeds, comparable to fast-flying modern birds. Meganeura's hunting strategy likely combined active pursuit with ambush tactics. It may have patrolled territories along swamp edges or over open water, searching for smaller insects with its exceptional vision. When prey was spotted, Meganeura would accelerate rapidly, using its large wings to close distance quickly. The final strike probably happened in mid-air, with Meganeura grabbing prey with its legs, each leg armed with spines for gripping, before delivering a killing bite with its mandibles. This aerial interception technique requires sophisticated neural processing to calculate trajectories and intercept angles, suggesting Meganeura had a relatively complex nervous system. The discovery of multiple Meganeura fossils in similar rock formations suggests they may have been common in their habitats. Some sites in France and England have yielded dozens of wing fragments from different individuals, indicating these giants weren't rare curiosities, but regular members of the ecosystem. Their abundance makes sense given the oxygen-rich atmosphere. The conditions that allowed them to exist also made them competitive and successful. Meganeura didn't live in isolation. The Carboniferous skies were filled with flying insects of many types, though none approached Meganeura's size. Smaller dragonfly relatives, early flying insects related to mayflies, and primitive winged forms all shared the airspace. Meganeura likely dominated this aerial community, taking prey from smaller species and facing little competition for the role of top aerial predator. On the ground below, giant millipedes and early amphibians posed no threat to something that spent most of its life in flight. Reproduction in Meganeura remains somewhat mysterious due to the lack of complete fossil specimens showing reproductive organs, but comparisons with modern dragonflies provide clues. Meganeura likely laid eggs in or near water, and the nymphs probably lived aquatic lives in the swamps. Modern dragonfly nymphs are voracious predators, and Meganeura nymphs were probably similar, though much larger. These aquatic juveniles may have fed on small fish, amphibian larvae, and other aquatic invertebrates, growing through multiple molts before finally emerging as winged adults. The transformation from aquatic nymph to aerial adult would have been a spectacular event. The nymph would have crawled out of the water onto a plant stem or rock, then split its exoskeleton along the back. The adult would have emerged soft and vulnerable, pumping fluid into its wings to expand them to full size. This process takes hours in modern dragonflies, and for something the size of Meganeura, it may have taken even longer. Once the exoskeleton hardened and the wings dried, the adult would have taken its first flight, joining the aerial ecosystem as one of its most formidable members. Meganeura's reign ended as oxygen levels declined. By the end of the Carboniferous, atmospheric oxygen began dropping from its peak, and giant insects started disappearing from the fossil record. Meganeura and its relatives vanished entirely, unable to sustain their large bodies as the atmosphere changed. Modern dragonflies, though impressive flyers, never approached the sizes their Carboniferous ancestors achieved. The 2-foot wingspan of Meganeura remains the upper limit of insect flight, a record set 300 million years ago and never broken since. Arthropleura, the 8-foot millipede. The forest floor of the Carboniferous was a dangerous place, but not because of what lurked in the shadows. It was dangerous because of what crawled openly across the leaf litter in broad daylight. Arthropleura was a millipede that defied every expectation of what arthropods could become. Reaching lengths of over 8 feet and estimated weights of 50 kilograms, it was the largest land invertebrate ever to exist. Picture a millipede as long as an alligator, as thick as a human torso, crawling through prehistoric forests on hundreds of legs, moving in coordinated waves. This wasn't a creature that hid from predators. It was too large to hide, too armoured to need to. The first Arthropleura fossils were discovered in the 1800s in European coal deposits, and they immediately posed problems for paleontologists. The fossils consisted mainly of articulated body segments, each one showing clear evidence of heavy armour plating. The segments locked together like overlapping shields, providing both flexibility and protection. Each segment bore a pair of leg attachments, and reconstructions based on trackways show that some individuals had more than 60 pairs of legs working in rhythmic coordination. What makes Arthropleura particularly fascinating is the completeness of some trace fossil evidence. In addition to body fossils, paleontologists have found trackways, preserved trails showing exactly how these giants moved. These trackways appear in Carboniferous rocks from North America and Europe, displaying parallel rows of footprints with spacing that matches the reconstructed body size. The trackways show that Arthropleura walked with a steady, wave-like gait, each leg stepping in precise sequence. The width of the trackways confirms that the largest individuals exceeded 8 feet in length, making them comparable in size to modern crocodiles. The body armour of Arthropleura was both defensive and structural. Each segment consisted of tough chitin reinforced with calcium carbonate, creating plates that could resist attacks from contemporary predators. The segments overlap slightly, allowing the animal to curl up if threatened, a defense mechanism still used by modern pill millipedes and pill bugs. When fully curled, Arthropleura would have presented nothing but armoured plates to any attacker, with no soft tissues exposed. This defensive posture would have made it nearly invulnerable to the predators of its time. But what did Arthropleura eat to fuel such a massive body? Unlike modern centipedes, which are predators, millipedes are generally detritivores, feeding on decaying plant matter. Arthropleura likely followed this pattern, consuming the abundant dead vegetation that accumulated in Carboniferous forests. The lack of lignin-degrading organisms meant that fallen trees and leaf litter piled up in thick layers, creating an endless food supply for large detritivores. Arthropleura's mouthparts show adaptations for chewing plant material rather than capturing prey, supporting the detritivore interpretation. The discovery of Arthropleura gut contents would provide definitive proof of diet, but such preservation is extremely rare. However, coprolites, fossilized feces, attributed to large arthropods, have been found in Carboniferous deposits. These coprolites contain fragments of plant material, including spores, woody tissue, and decomposed vegetation. While not all coprolites can be definitively linked to Arthropleura, their size and contents match what would be expected from a giant detritivorous millipede. Arthropleura's respiratory system was the key to its size. Like all arthropods, it breathed through spiracles, small openings along the sides of its body that led to tracheal tubes. In the oxygen-rich Carboniferous atmosphere, these tubes could efficiently supply oxygen to tissues deep within the body. Mathematical models of arthropod respiration show that at 35% oxygen, tracheal systems can support bodies much larger than possible in modern atmospheric conditions. Arthropleura pushed this system to its absolute limits, achieving sizes that would cause respiratory failure in today's atmosphere. The habitat preferences of Arthropleura can be inferred from where fossils are found. Most specimens come from deposits that represent ancient swamp forests, waterlogged environments with abundant dead vegetation. These swamps provided everything Arthropleura needed: food in the form of plant debris, humidity to prevent desiccation, and shelter among the massive fallen trees. The warm, stable climate of the Carboniferous tropics meant these swamps persisted year-round, supporting populations of giant arthropods without seasonal disruptions. Reproduction in Arthropleura remains largely unknown due to the lack of preserved reproductive structures. Modern millipedes typically lay eggs in soil or rotting wood, with the young hatching as miniature versions of adults and growing through successive molts. Arthropleura probably followed a similar pattern, though the eggs would have been proportionally larger. Finding a clutch of Arthropleura eggs would be an extraordinary discovery, but the acidic conditions of swamps rarely preserve such delicate structures. Predation pressure on adult Arthropleura was likely minimal due to its size and armour. The largest predators of the Carboniferous land environments were early amphibians and arachnids, none of which approached Arthropleura in size. A fully grown Arthropleura would have been effectively immune to predation, occupying the role of a mega-herbivore in modern ecosystems, too large and too well defended to be worth attacking. Juveniles would have been more vulnerable, but rapid growth probably moved them quickly into size classes that predators couldn't handle. The extinction of Arthropleura coincided with the decline in atmospheric oxygen at the end of the Carboniferous. As oxygen levels dropped, the maximum sustainable size for tracheal-breathing arthropods decreased. Arthropleura and similar giant forms disappeared from the fossil record, replaced by smaller relatives that could survive in the changing atmospheric conditions. No millipede since has approached even half the length of Arthropleura, and none likely ever will unless Earth's atmosphere undergoes another oxygen revolution. Pulmonoscorpius, the giant scorpion. Darkness falls in the Carboniferous forest, and the real hunting begins. Pulmonoscorpius emerges from beneath a rotting log, its body measuring over 2 feet long, with pincers that could crush smaller arthropods and a tail armed with a venomous stinger the size of a human thumb. This wasn't a creature to stumble across in the night. It was a fully equipped predator, combining size, armor, venom, and sensory capabilities that made it one of the most dangerous hunters on the Carboniferous forest floor. Pulmonoscorpius kirktonensis was first described from fossils found in Scotland, in rocks dating to the early Carboniferous. The specimens were remarkably complete, preserving not just the hard exoskeleton, but also traces of internal structures including book lungs, the respiratory organs that give this scorpion its scientific name. These book lungs were substantially larger than those of modern scorpions, allowing Pulmonoscorpius to grow to sizes that would be impossible for scorpions breathing today's air. The anatomy of Pulmonoscorpius follows the standard scorpion body plan, but scales it up dramatically. The pedipalps, the large pincers at the front, were powerful crushing tools capable of seizing and holding prey. Each pincer had a movable finger and fixed finger that worked like pliers, with enough force to crack open the exoskeletons of other arthropods. The surface of the pincers shows areas of heavy wear in some fossils, indicating they were used extensively to process hard-bodied prey. Behind the pincers, 4 pairs of walking legs provided stable locomotion across the uneven forest floor. Scorpions are ambush predators that wait for prey to come within striking distance, but they also actively hunt when necessary. Pulmonoscorpius likely did both, using different strategies depending on circumstances. Its large size would have made extended pursuits energetically expensive, favouring ambush tactics. But when hunting smaller or slower prey, active stalking would have been effective. The tail of Pulmonoscorpius was its signature weapon. Scorpion tails consist of 5 segments ending in a bulbous telson that contains venom glands. In Pulmonoscorpius, this telson was massive, with a sharp, curved stinger that could penetrate deep into prey. The venom produced by Pulmonoscorpius is unknown. Venom doesn't fossilize. But comparisons with modern scorpions suggest it was probably neurotoxic, causing paralysis and death in prey. Whether it was dangerous to humans is impossible to say, but given the size of the stinger and the likely potency needed to subdue large prey, caution would have been wise. Scorpions detect prey primarily through vibrations rather than vision. They have numerous sensory hairs covering their legs and pedipalps that can detect the slightest movement in the substrate. Pulmonoscorpius would have felt the footsteps of approaching prey through these sensors, allowing it to ambush animals in complete darkness. The Carboniferous nights were genuinely dark. No moon to speak of in the way we have today, and the thick forest canopy would have blocked most starlight. In this environment, vibration-based hunting gave scorpions a significant advantage. Evidence for Pulmonoscorpius's diet comes from examining the prey available in its environment. The Carboniferous ecosystem was rich with potential food sources: smaller scorpions, large insects, millipedes, early terrestrial arthropods of many types, and possibly small amphibians. Some researchers suggest Pulmonoscorpius may have occasionally scavenged, feeding on dead animals it encountered. The powerful pincers could tear apart carcasses, and scorpions have strong digestive enzymes capable of breaking down tough tissues. One particularly interesting aspect of Pulmonoscorpius is its respiratory system. Book lungs are internal structures where air circulates over stacked, plate-like chambers rich in blood vessels. This design allows efficient gas exchange, but limits body size in modern scorpions due to the difficulty of moving oxygen through the system. In the oxygen-rich Carboniferous atmosphere, book lungs could support much larger bodies. The preserved book lung structures in Pulmonoscorpius fossils show they were proportionally larger than in modern scorpions, with more lamellae, the individual plates where gas exchange occurs. Pulmonoscorpius likely faced competition from other predators. Large spiders also inhabited the Carboniferous forests, along with early amphibians that ventured onto land to hunt. The division of ecological niches probably separated these predators: scorpions hunting at night and in dense undergrowth, amphibians active near water, and spiders using webs in more open areas. This niche partitioning would have reduced direct competition and allowed multiple predator types to coexist. Reproduction in scorpions is complex and would have been similar in Pulmonoscorpius. Modern scorpions engage in elaborate courtship dances where the male and female grasp pincers and move back and forth before mating. After mating, females give birth to live young that climb onto the mother's back and remain there for protection during their first molt. If Pulmonoscorpius followed this pattern, seeing a 2-foot scorpion carrying dozens of miniature scorpions on its back would have been quite a sight. The fossil record shows that Pulmonoscorpius and similar giant scorpions disappeared at the end of the Carboniferous as oxygen levels declined. Modern scorpions, though still impressive predators in their ecosystems, are limited to much smaller sizes. The largest living scorpion barely reaches 8 inches, less than half the length of Pulmonoscorpius. The age of giant scorpions was brief in geological terms, lasting perhaps 30 million years before changing atmospheric conditions made such sizes unsustainable. Meganuropsis, the Permian super predator. If Meganeura was the undisputed ruler of Carboniferous skies, its descendant Meganuropsis took that crown and wore it even larger. Living in the early Permian period, Meganuropsis permiana represents the absolute pinnacle of insect flight evolution. With wingspans potentially exceeding 75 centimeters, nearly 30 inches, it was even larger than its famous predecessor. This wasn't just an incremental increase. Meganuropsis represented the final, ultimate expression of what high oxygen levels could create in a flying insect. The fossils of Meganuropsis were discovered in Permian rocks from Kansas, preserved in fine-grained shale that captured wing details with stunning clarity. When these specimens were first studied, they revealed wing structures that surpassed all known dragonfly relatives in both size and complexity. The wings showed advanced venation patterns with reinforced leading edges and areas of specialized membrane that likely improved flight efficiency. These adaptations suggest Meganuropsis was not just large, but also a highly sophisticated flyer. The head of Meganuropsis, based on fragments and comparison with related species, was dominated by massive compound eyes that would have provided exceptional visual acuity. These eyes likely contained even more lenses than Meganeura's eyes, allowing Meganuropsis to detect prey from greater distances and track multiple targets simultaneously. The neural processing required for this level of visual sophistication suggests a relatively large brain for an insect, with specialized regions dedicated to visual processing and flight control. The thorax of Meganuropsis was a powerhouse of muscle tissue. Flying at large sizes requires substantial muscle mass to generate the lift and thrust needed to stay airborne. The flight muscles of Meganuropsis would have filled much of the thorax, with attachments to the wing bases providing mechanical advantage for the wing strokes. Calculations based on wing area and estimated body mass suggest that Meganuropsis operated near the theoretical limits of insect flight mechanics. Any larger, and the wings wouldn't have been able to generate enough lift. Meganuropsis lived during a time when oxygen levels were still elevated, though beginning their long decline from Carboniferous peaks. The early Permian atmosphere contained approximately 30% oxygen, still well above modern levels, but lower than the Carboniferous maximum. This slightly reduced oxygen concentration may have represented the absolute minimum needed to support insects of Meganuropsis' size, explaining why even larger forms never evolved. The hunting strategy of Meganuropsis can be inferred from its morphology and the prey available in its environment. The Permian landscape was home to numerous flying insects, though none approached Meganuropsis in size. Early beetles, primitive flying insects related to modern mayflies, and smaller dragonfly relatives would all have been potential prey. Meganuropsis likely hunted using a combination of endurance flight and rapid acceleration, cruising over lakes and wetlands while scanning for prey below, then diving at high speed to intercept targets. The capture mechanism probably involved precise aerial interception. Meganuropsis' legs were equipped with rows of sharp spines that could lock together to form a catching basket. As it closed on prey, it would swing its legs forward, forming a trap that snared the victim in mid-flight. The momentum of the strike, combined with the spines, would ensure the prey couldn't escape. Once secured, Meganuropsis would have used its powerful mandibles to deliver a killing bite before consuming the prey in flight or landing to feed. Evidence for Meganuropsis behavior also comes from the environments where fossils are found. The Kansas deposits represent ancient lake shores and wetland margins, exactly the habitats modern dragonflies prefer. These areas would have provided abundant prey, including both aerial insects and emergent adults from aquatic larvae. The combination of open water for flight space and vegetated margins for perching made these environments ideal for large aerial predators. The life cycle of Meganuropsis almost certainly included an aquatic larval stage, following the pattern of all dragonflies and their relatives. The larvae would have lived in Permian lakes and ponds, developing through multiple instars before finally emerging as adults. These aquatic nymphs were likely fearsome predators themselves, hunting fish, amphibian larvae, and other aquatic invertebrates. The final emergence, when the adult crawled from the water and underwent metamorphosis, would have been a vulnerable period requiring safe emergence sites away from predators. Competition among Meganuropsis individuals may have been intense, particularly for territory. Modern dragonflies are highly territorial, with males defending hunting grounds and breeding sites from rivals. Given the large size and energy requirements of Meganuropsis, territories would have needed to be substantial to provide enough prey. Aerial battles between rival males could have been spectacular, with two giant dragonflies grappling in mid-air, each trying to drive the other from contested airspace. The extinction of Meganuropsis came as atmospheric oxygen continued its decline through the Permian. By the end of the period, oxygen levels had dropped below the threshold needed to support such large flying insects. The Great Permian Extinction Event, which devastated life on Earth, also marked the end of the giant dragonfly era. After Meganuropsis, no flying insect ever achieved comparable size. The laws of physics and chemistry that had briefly allowed insects to dominate the skies reasserted their limits, and flying insects returned to more modest proportions. The forest canopy ecosystem. Above the Carboniferous forest floor, a three-dimensional world existed in layers, each with its own community of organisms adapted to specific heights and conditions. The canopy ecosystem of the Carboniferous was radically different from modern forests, populated by arthropods that filled every niche from bark boring to leaf mining to aerial predation. Understanding this vertical stratification reveals how complex these ancient ecosystems were and how thoroughly insects had colonized every available habitat. The uppermost canopy, where the tallest scaled trees reached heights exceeding 100 feet, was a realm of extremes. Here the air was fresher, with higher oxygen concentrations due to proximity to actively photosynthesizing leaves. The temperature fluctuated more dramatically than in the understory, with intense heat during the day and cooler conditions at night. This zone was dominated by flying insects that could handle the open air and strong winds. Meganuropsis and Meganeura patrolled these heights, hunting smaller flying insects that emerged from lower levels. Below the highest canopy, at middle heights perhaps 50 to 70 feet up, a different community existed. This zone had moderate light penetration and more stable temperature conditions. Here lived insects specialized for living on bark and among the fronds of tree ferns. Some early insects had evolved the ability to bore into bark, creating tunnels where they could feed on nutrient-rich cambium layers. These bark-boring insects were crucial members of the ecosystem, starting the process of breaking down living wood and creating entrance points for decay organisms. The lower canopy and understory, from ground level to perhaps 30 feet high, was the most densely populated zone. This level received filtered light through the upper canopy, creating a twilight environment even during the day. Humidity levels were highest here, approaching saturation in many areas due to the constant transpiration from vegetation and evaporation from swamp waters. This humid, shaded environment was perfect for arthropods that needed to avoid desiccation. Within this lower zone lived an incredible diversity of arthropods. Primitive wingless insects scurried along tree trunks and through leaf litter. Early relatives of modern bristletails and silverfish lived under bark and in rotting logs. These ancient insects were among the first to colonize land, and they thrived in the humid Carboniferous environment. Their small size and ability to squeeze into tight spaces allowed them to exploit microhabitats unavailable to larger organisms. Spiders were important predators in this lower canopy zone. While not as gigantic as scorpions or millipedes, Carboniferous spiders were still larger than most modern species, and they employed the same successful strategy their descendants use today: webs. These early webs were probably simple sheet webs, rather than the elaborate orb webs some modern spiders construct, but they were effective at capturing flying insects in the cluttered environment of the understory. Plant-feeding insects occupied specialized niches throughout all canopy levels. Some fed on tree fern fronds, others on the scale-like leaves of lepidodendron trees, and still others on the foliage of seed ferns. These herbivorous insects showed early adaptations for dealing with plant defenses. Some plants had begun producing toxic compounds to deter herbivory, and insects responded by evolving resistance to these toxins, or specializing on particular plant species. Evidence for this plant-insect interaction comes from fossil leaves showing characteristic damage patterns. Paleobotanists have identified leaves with holes chewed by mandibulate insects, margins showing edge-feeding damage, and even traces of leaf mining, where insect larvae tunnel between the upper and lower surfaces of leaves. These damage patterns become more common and diverse through the Carboniferous, documenting an escalating evolutionary arms race between plants and their insect herbivores. The vertical stratification extended even to the forest floor and below ground. Root systems of Carboniferous trees were often shallow and extensive, creating networks near the surface. Insects and other arthropods colonized this underground frontier, feeding on roots, fungal hyphae, and decaying plant matter. Some early insects were almost certainly root feeders, using mouthparts adapted for chewing through tough root tissue to access nutrient-rich fluids inside. Decomposition was a critical process in the Carboniferous ecosystem, and arthropods played a central role. While lignin-degrading fungi had not yet evolved, mechanical breakdown of plant material by arthropods was essential for nutrient cycling. Millipedes like Arthropleura consumed dead plant matter, breaking it into smaller pieces and passing it through their digestive systems. Their feces became substrate for other decomposers, including bacteria and early fungi that could process simpler organic compounds. The Canopy ecosystem also included important abiotic factors that shaped where organisms could live. Wind patterns differed between canopy levels, with stronger, more consistent winds in the upper canopy and more stagnant air below. This affected flying insects, which had to compensate for wind when navigating. Rainfall behaved differently at various heights, with the upper canopy intercepting much of the precipitation and creating a drip pattern in lower levels. These microhabitat variations created a mosaic of conditions that different species exploited. Seasonal changes, though less pronounced in the tropical Carboniferous climate than in temperate modern forests, still occurred. Evidence from growth rings in fossil wood shows that even tropical Carboniferous forests experience some seasonality, possibly related to monsoon patterns. During wetter periods, the canopy would have been more active with increased plant growth and insect reproduction. Drier periods may have triggered migrations of some insects to more favourable areas or induced dormancy in others. The complexity of the Carboniferous canopy ecosystem rivals modern rainforests in its vertical stratification and specialisation. Each level supported distinct communities adapted to its specific conditions. This three-dimensional habitat structure allowed an extraordinary diversity of arthropods to coexist by dividing resources vertically as well as horizontally. The result was an ecosystem of remarkable productivity and complexity, driven primarily by arthropod activity and the high oxygen atmosphere that made their success possible. Euphoberia, the armoured millipede. Walking the margins between water and land, where the swamp forest transitioned to drier ground, lived a millipede that looked like it was preparing for battle. Euphoberia wasn't as large as Arthropleura. Few organisms were, but it compensated for smaller size with spectacular defensive adaptations. Each body segment bore not just armour plates, but also prominent spines that projected outward and backward, creating a living fortress that predators learned to avoid. Euphoberia fossils have been found across North America and Europe in Carboniferous deposits, often preserved in remarkable detail that shows individual spines and segment articulation. The body length typically ranged from 8 to 12 inches, respectable for a millipede but modest compared to the giants of the period. What distinguished Euphoberia wasn't size, but the elaborate defensive architecture covering its body. The spines on each segment were hollow, filled with glands that produced defensive chemicals. Modern millipedes use a similar strategy, secreting toxic or noxious compounds from glands along their sides to deter predators. Euphoberia took this defense to an extreme, with spines that could deliver these chemicals directly to anything that touched them. Some researchers suggest the spines may have been capable of injecting venom like modern scolopendrid centipedes, though this remains speculative without direct evidence. The chemical composition of Euphoberia's defensive secretions is unknown. Organic compounds don't preserve in fossils, but comparisons with modern millipedes provide clues. Living millipedes produce a remarkable variety of defensive chemicals, including benzoquinones, hydrogen cyanide, phenols, and alkaloids. These compounds can burn skin, repel predators, or even be toxic if ingested. Euphoberia likely produce similar or even more potent compounds, given the selection pressure from large predators in its environment. The armoured segments of Euphoberia overlapped in a way that allowed flexibility while maintaining protection. Each segment could move independently, allowing the animal to navigate over uneven terrain or curl into a defensive ball. When threatened, Euphoberia would have coiled tightly, presenting only spines and armoured plates to attackers. This defensive posture, combined with chemical secretions, would have made it an unpalatable target for most predators. Euphoberia's habitat preferences can be inferred from where fossils are found. Most specimens come from deposits representing forest margins and drier upland areas rather than the wettest swamp environments. This suggests Euphoberia may have preferred slightly better drained soils, where leaf litter accumulated in thick layers, but standing water was less common. These transitional zones between wet swamps and drier forests would have offered abundant food in the form of decaying vegetation while avoiding the most waterlogged conditions. The diet of Euphoberia was almost certainly detritivorous, feeding on dead plant matter like most millipedes. The mouthparts show adaptations for chewing and grinding rather than capturing live prey. Euphoberia would have consumed fallen leaves, decaying wood, and decomposing plant debris, playing a crucial role in nutrient cycling. Its digestive system would have contained symbiotic microorganisms that help break down cellulose and other complex plant compounds. Reproduction in Euphoberia probably followed patterns similar to modern millipedes. Males would have sought out females, possibly using chemical signals, pheromones, to locate potential mates. Mating in millipedes often involves the male depositing a packet of sperm that the female picks up, a process that can take hours and involves elaborate positioning and tactile communication. After mating, females would have laid eggs in protected locations, possibly in burrows or under rotting logs. The juveniles that hatch from these eggs would have been miniature versions of adults, though with fewer segments. Millipedes grow by adding segments with each molt, gradually increasing in length through successive stages. Young Euphoberia would have been vulnerable to predators despite their spines, making growth to adult size crucial for survival. The high oxygen atmosphere allowed rapid growth, with abundant food resources supporting the energetic demands of frequent molting. Euphoberia faced predation pressure primarily from large amphibians and possibly early reptiles that ventured onto land during the Carboniferous. These vertebrate predators had jaws powerful enough to potentially crush millipede armor, but the spines and chemical defenses made Euphoberia a challenging target. Predators that attempted to eat Euphoberia likely learned quickly that the experience was unpleasant, creating a learned avoidance that protected the millipede population. The evolutionary success of Euphoberia's defensive strategy is evident in its wide distribution. Finding fossils across multiple continents suggests this millipede was common and successful throughout the Carboniferous period. The combination of armor, spines, and chemical defenses represents a peak in millipede defensive evolution, a strategy so effective that it allowed Euphoberia to thrive for millions of years. When oxygen levels declined at the end of the Carboniferous, Euphoberia faced the same challenges as other large arthropods. Its lineage eventually went extinct, though millipedes themselves survived and continue to thrive today. Modern millipedes, while much smaller, still employ many of the same defensive strategies, armored segments, defensive postures, and chemical secretions that made Euphoberia successful. The spine millipedes of the Carboniferous represent an extreme version of defensive adaptations that still work effectively on a smaller scale in modern ecosystems. The aquatic giants. While terrestrial and aerial insects captured the spotlight during the Carboniferous, the aquatic realm harbored its own giants. The extensive swamp systems and permanent bodies of fresh water supported diverse communities of aquatic arthropods that rivaled their terrestrial relatives in size and ferocity. These underwater ecosystems were complex, layered environments where dragonfly larvae, giant water scorpions, and enormous aquatic insects created food webs as intricate as those on land. The nymphs of Meganeura and Meganuropsis were fearsome aquatic predators in their own right. These immature forms lived entirely underwater, breathing through internal gills and hunting with extendable mouthparts called labia. Modern dragonfly nymphs use a similar structure, a hinged lower lip that can shoot forward to grab prey in a fraction of a second. The Carboniferous giants would have had proportionally larger labium capable of snatching fish-sized prey. These nymphs occupied the role of apex predators in many aquatic systems. Their development from egg to adult spanned multiple years, possibly 5 to 7 years for the largest species, during which they molted repeatedly and increased in size dramatically. Each molt represented a vulnerable period when the new exoskeleton was soft, but it also allowed for growth spurts that moved the nymph into larger size classes less vulnerable to predation. Fossil evidence for these aquatic nymphs is surprisingly good. Several sites preserved nymphs alongside adult specimens, allowing paleontologists to link immature and adult forms. These fossils show body plans optimized for underwater ambush hunting, low profiles, powerful legs for clinging to submerged vegetation, and robust heads with prominent eyes. The fossil nymphs reached lengths exceeding 6 inches, confirming that these were substantial predators capable of taking sizable prey. Competition in the aquatic environment came from other predatory arthropods, including early water scorpions that grew to impressive sizes. Though not true scorpions, these aquatic hemipterans evolved raptorial front legs and breathing snorkels that allowed them to hunt near the water surface. Some Carboniferous water scorpions exceeded a foot in length, making them formidable predators of smaller fish and aquatic insects. The prey base supporting these aquatic giants consisted of smaller insects, crustaceans, early fish, and amphibian larvae. Carboniferous waters contain diverse fish populations, including early ray-finned fish, lobe-finned fish related to modern coelacanths and lungfish, and heavily armoured plaxiderms that had survived from the Devonian. These fish provided abundant food for predatory nymphs, though the larger fish could also prey on smaller nymphs, creating reciprocal predation dynamics. Amphibian larvae added another layer to aquatic food webs. The Carboniferous saw major diversification of amphibians, with numerous lineages adapted to aquatic lifestyles. Many amphibians retained an aquatic larval stage before metamorphosing into terrestrial adults. And these larvae were both predators and prey in the underwater ecosystem. Large dragonfly nymphs could prey on small amphibian larvae, while larger amphibians could consume smaller nymphs. Oxygen levels in Carboniferous waters were likely higher than modern freshwater systems due to the elevated atmospheric oxygen. This would have supported higher metabolic rates in aquatic organisms and allowed larger body sizes among water-breathing arthropods. The extensive vegetation in and around water bodies also contributed to oxygen production through photosynthesis, creating well-oxygenated conditions even in relatively stagnant swamp waters. Aquatic plants during the Carboniferous were diverse and provided essential habitat structure. Submerged vegetation offered hiding places for prey and ambush sites for predators. Emergent vegetation, plants growing up from the water, provided perches where adult insects could rest and where nymphs could climb during metamorphosis. Floating vegetation created surface habitat where some insects laid eggs and where surface-dwelling arthropods hunted. The transition from water to air, the emergence of adult flying insects from aquatic nymphs, was a critical moment in the life cycle. This emergence had to be timed correctly, typically occurring at night or early morning when temperatures were lower and humidity higher to prevent desiccation. The newly emerged adult was soft and vulnerable for several hours while its wings expanded and hardened, making emergent sites crucial. Vegetation near water provided these essential emergent sites, and their availability may have limited populations in some areas. Evidence for emergence behavior comes from fossil sites that preserve both nymphs and adults together, suggesting these were areas where emergence commonly occurred. Some fossils even capture the moment of emergence, with shed-nymphal exoskeletons still clinging to plant stems. These rare fossils provide direct evidence of the transformation from aquatic predator to aerial hunter. The aquatic ecosystems of the Carboniferous were remarkably productive, supporting dense populations of predators and prey. The combination of high oxygen levels, warm temperatures, abundant nutrients from decaying vegetation, and extensive habitat created ideal conditions for aquatic arthropods. These systems were in some ways more stable than terrestrial environments, buffered against temperature extremes and providing year-round hunting opportunities for predators. Mazothairos, the giant water bug. If dragonfly nymphs were the lions of Carboniferous waters, then Mazothairos was the crocodile, an even larger, more powerful predator that lurked in deeper areas waiting for substantial prey. This enormous hemipteran, related to modern giant water bugs, exceeded 10 inches in length and possessed raptorial front legs modified into powerful grasping weapons. The discovery of Mazothairos remains some of the most impressive evidence of how large aquatic insects grew during the age of high oxygen. The fossils of Mazothairos come from Carboniferous deposits in France, preserved in fine-grained lake sediments that captured exceptional detail. The front legs show the characteristic adaptations of ambush predators. Thick, powerful segments with rows of sharp spines that could lock together to form an inescapable trap. These legs could snap shut with remarkable speed, impaling prey on the spines before dragging it toward the mouthparts. Modern giant water bugs, though much smaller than Mazothairos, are among the most voracious aquatic predators alive. They hunt fish, frogs, snakes, and even small turtles, using their front legs to capture prey and their piercing mouthparts to inject digestive enzymes. Mazothairos almost certainly employed similar tactics, but at a much larger scale. Its prey would have included fish up to several inches long, amphibian larvae, and other aquatic insects, including smaller individuals of its own species. The mouthparts of Mazothairos consisted of a sharp, needle-like beak that could pierce prey and inject both toxins and digestive enzymes. This extraoral digestion, liquefying prey tissues outside the body before sucking them in, allowed Mazothairos to consume prey larger than its mouth could otherwise accommodate. The process was probably slow, with Mazothairos holding captured prey for extended periods, while enzymes broke down internal tissues. Breathing presented challenges for large aquatic insects. Modern giant water bugs solved this by carrying air bubbles trapped against their bodies, periodically surfacing to refresh their air supply. Mazothairos likely used a similar system, but enhanced by the high oxygen content of Carboniferous air. Each breath captured more oxygen than would be possible today, extending the time Mazothairos could remain submerged between trips to the surface. The habitat of Mazothairos was probably the deeper, more permanent water bodies within the swamp system, lakes and ponds that maintained consistent water levels year-round. These environments provided stable hunting grounds where Mazothairos could establish territories and ambush prey repeatedly. The dark waters of Carboniferous swamps would have provided excellent concealment for an ambush predator, with prey unable to see the hunter until the attack was already underway. Sexual dimorphism, physical differences between males and females, was probably present in Mazothairos, though the fossil record doesn't provide definitive evidence. Modern water bugs show size differences between sexes, and some species practice male parental care, with males carrying eggs on their backs until they hatch. Whether Mazothairos exhibited similar behavior remains unknown, but it suggests possibilities for complex reproductive biology even in giant aquatic insects. The life cycle of Mazothairos would have been entirely aquatic, without the dramatic metamorphosis seen in dragonflies. Hemipterans undergo incomplete metamorphosis, with nymphs resembling small adults and growing through successive molts. Young Mazothairos would have hatched from eggs as miniature predators, hunting progressively larger prey as they grew. This gradual development meant that individuals of different sizes occupied different ecological niches, reducing competition between age classes. Predation on Mazothairos would have come primarily from larger fish and aquatic amphibians. Despite its formidable weaponry, Mazothairos was not invulnerable. Large predatory fish could swallow smaller individuals whole, while mature amphibians might have hunted adult Mazothairos in surprise attacks. The balance between Mazothairos and its potential predators would have created a dynamic ecosystem where size and behavior determined survival. The extinction of Mazothairos lineage, like other giant insects, came with declining oxygen levels. Modern giant water bugs, though impressive predators, reach maximum lengths of only 4 inches, less than half the size of Mazothairos. The physics of respiration in aquatic environments, where oxygen must be obtained from air rather than dissolved in water for insects breathing through trachea, set absolute limits on size that vary with atmospheric oxygen content. The discovery of Mazothairos expanded our understanding of how thoroughly insects dominated Carboniferous ecosystems. Not only did they rule the skies and the forest floor, but they also commanded the waters, filling the apex predator role in aquatic food webs. This complete dominance across all habitats represents a unique period in Earth's history when insects achieved a level of ecological importance unmatched before or since. The smaller predators. While giants like Meganeura and Arthropleura capture imagination, the Carboniferous ecosystem was filled with smaller but equally important predators. These arthropods, ranging from a few millimeters to a few inches in length, occupied every imaginable niche and created the complex food web foundation that supported the giants. Understanding these smaller predators reveals the true diversity and ecological sophistication of the age of insects. Centipedes were among the most important small to medium predators in the Carboniferous. Unlike their millipede relatives, centipedes are carnivorous, hunting other arthropods with speed and venom. Carboniferous centipedes grew larger than most modern species, with some reaching lengths of 8 to 12 inches. Their bodies were segmented, with each segment bearing a pair of legs. The more segments, the more legs, with some species having over 100 pairs. The first pair of legs in centipedes are modified into fangs called forcipules. These hollow structures connect to venom glands and function like hypodermic needles, injecting venom that paralyzes prey. The venom composition likely varied among species, with some producing neurotoxins that cause rapid paralysis and others using slower-acting compounds. Fossil centipedes show well-developed forcipules, confirming they were active predators in Carboniferous ecosystems. Hunting strategies in centipedes rely on speed and surprise. Modern centipedes are among the fastest arthropods for their size, capable of rapid bursts of locomotion when pursuing prey or fleeing predators. Carboniferous centipedes probably employed similar tactics, hiding under bark or in leaf litter during the day and actively hunting at night. Their numerous legs provided exceptional traction on irregular surfaces, allowing them to chase prey over bark, through vegetation, or across the forest floor. Spiders represented another major group of small predators. While not as gigantic as some other Carboniferous arthropods, spiders during this period were larger and more diverse than their earlier ancestors. The evolution of silk production gave spiders a tremendous advantage, allowing them to construct webs that passively captured prey. Carboniferous spider webs were probably simpler than modern orb webs, consisting of sheets or tangles rather than the elaborate geometric patterns some modern spiders create. The prey captured by spider webs consisted primarily of flying insects. As flying insects diversified and became more abundant during the Carboniferous, spiders that could exploit this aerial food source gained significant advantages. Webs stretched between vegetation could intercept insects moving through the forest at various heights. The spider would detect vibrations when an insect struck the web, then rush out to bite and wrap the prey in silk before it could escape. Evidence for spider webs in the Carboniferous comes primarily from the presence of silk-producing structures in fossil spiders. The spinnerets, organs at the tip of the abdomen that produce silk, are sometimes preserved, showing that Carboniferous spiders possess this capability. Direct evidence of webs themselves is virtually non-existent, as silk rarely fossilizes, but the anatomical evidence leaves little doubt that web construction was already well established. Predatory mites and other small arachnids filled microhabitat niches too small for larger predators. These tiny arthropods, often less than a millimeter in length, hunted even smaller organisms, including springtails, early insect larvae, and other mites. Despite their size, these micropredators were crucial ecosystem members, controlling populations of decomposers and helping regulate nutrient cycling at the smaller scales. The diversity of predatory strategies among smaller arthropods was remarkable. Some employed active hunting, constantly moving in search of prey. Others used sit-and-wait tactics, remaining motionless until prey came within striking distance. Still others employed specialized techniques like trap setting, where silk or carefully positioned body parts created mechanisms to capture prey. This variety of strategies allowed multiple predator species to coexist by exploiting different prey types and using different hunting methods. Competition among small predators would have been intense in areas with high prey density. Territoriality probably evolved in some species, with individuals defending productive hunting areas from conspecifics. Other species may have avoided competition through temporal niche partitioning, hunting at different times of day or night, when other predators were less active. Still others specialized on particular prey types that competitors couldn't efficiently exploit. The relationship between small predators and the ecosystem's giants was complex. Small predators served as prey for larger species, transferring energy from the decomposer level up through the food web. But small predators also competed with large predators in some contexts, particularly when hunting similar prey. A centipede and a small scorpion might both hunt the same ground-dwelling insects, creating competition despite their size difference. Small predators also played roles in controlling pest populations. Herbivorous insects that damaged living plants could reach outbreak levels without predation pressure. Small predators kept these herbivore populations in check, preventing any single species from completely defoliating plants or causing ecosystem-wide damage. This regulatory role was essential for maintaining ecosystem stability and diversity. The fossil record of small predators is less complete than for giants, simply because smaller organisms are less likely to fossilize and more difficult to find. However, exceptional preservation sites occasionally capture assemblages of small arthropods, providing windows into this hidden diversity. Sites with fine-grained sediments that settle quickly in calm water sometimes preserve tiny predators in perfect detail, showing morphological adaptations and even gut contents in rare cases. The herbivore revolution. While predators often dominate discussions of ancient ecosystems, the Carboniferous witnessed an equally important revolution in herbivory. Early land plants had faced minimal browsing pressure from animals, but during the Carboniferous, insects evolved numerous strategies for feeding on living plant tissue. This herbivore revolution transformed plant-insect interactions and set the stage for the complex co-evolutionary relationships that define modern ecosystems. The earliest terrestrial insects were probably detritivores, or general feeders, consuming dead plant material or fungi rather than attacking living plants. But as plants diversified and became more abundant during the Carboniferous, natural selection favoured insects that could exploit this rich food source. The evolution of herbivory required overcoming significant challenges, particularly the chemical and physical defences plants employed to deter feeding. Physical defenses in Carboniferous plants included tough outer layers, silica-reinforced tissues, and sharp trichomes, hair-like structures that made surfaces difficult to navigate. Some plants developed thorns or spines on stems and leaves. These mechanical defenses were effective against casual browsing, but could be overcome by insects with specialized mouthparts. Mandibulate insects, those with chewing mouthparts, evolved stronger jaws capable of cutting through tough plant tissues. Chemical defenses presented a more sophisticated challenge. Plants produce a vast array of secondary metabolites, compounds not essential for basic metabolism but effective at deterring herbivores. These include tannins, phenolic compounds, alkaloids, and terpenoids, among many others. These chemicals can be toxic, distasteful, or digestive inhibitors. Carboniferous plants were already producing some of these compounds, creating selection pressure for insects to evolve resistance or avoidance strategies. Insect responses to plant chemical defenses took multiple forms. Some insects evolved enzymes capable of breaking down or neutralizing plant toxins. Others developed behaviors to avoid the most toxic plant tissues, feeding only on specific parts where defensive compounds were less concentrated. A particularly successful strategy was specialization, focusing on a single plant species or family and evolving specific adaptations to overcome that plant's unique defenses. Evidence for herbivory in the Carboniferous comes from fossil leaves showing characteristic damage patterns. Paleobotanists have documented leaves with holes from chewing, margins showing edge feeding, and even the distinctive trails left by leaf-mining larvae that tunnel between leaf surfaces. These damage patterns increase in frequency and diversity through the Carboniferous, documenting the escalating interaction between plants and herbivorous insects. One of the most significant evolutionary innovations was the ability to feed on vascular tissue, the xylem and phloem that transport water and nutrients through plants. Piercing sucking insects evolved mouthparts modified into a sharp beak that could penetrate plant epidermis and reach vascular tissue. This feeding strategy provided access to nutrient-rich fluids, while avoiding many of the physical and chemical defenses concentrated in outer tissues. The evolution of complete metamorphosis in some insect lineages, where larvae look completely different from adults, may have been partly driven by herbivory. This life history strategy allows larvae and adults to exploit different food sources, reducing competition between life stages. Herbivorous larvae could specialize on leaves, while adults fed on nectar, pollen, or didn't feed at all, creating efficiencies that benefited the overall population. Gall formation represents a particularly sophisticated form of herbivory. Some insects evolved the ability to chemically manipulate plant growth, causing plants to produce abnormal structures called galls that provided shelter and food for developing insect larvae. Evidence for galls appears in the Carboniferous fossil record, showing that this complex interaction evolved surprisingly early in the history of plant-insect relationships. The impact of herbivory on Carboniferous ecosystems was substantial. By consuming living plant tissue, herbivorous insects transferred energy from primary producers directly to higher trophic levels. This energy pathway was more efficient than the detritivore route, where nutrients had to wait for plant death and decomposition. The abundance of herbivorous insects supported larger populations of predators, increasing overall ecosystem complexity. Plants responded to herbivore pressure with evolutionary innovations of their own. The Carboniferous saw increased diversity in plant defensive compounds and structures. Some plants evolved rapid regrowth capabilities, allowing them to recover quickly from herbivore damage. Others developed induced defenses, defensive compounds produced only after damage occurred, saving energy when herbivores were absent. The co-evolutionary dance between plants and herbivorous insects accelerated through the Carboniferous. Each plant innovation in defense selected for insect counter-adaptations, which in turn selected for improved plant defenses. This reciprocal selection created a biological arms race that drove diversification in both groups and established patterns of interaction that continue in modern ecosystems. The herbivore revolution also affected nutrient cycling. By consuming living tissue and excreting partially digested material, herbivorous insects altered the chemical composition and decomposition rates of plant litter. Their feeding damage created entry points for pathogens and decomposers, accelerating breakdown of plant material. This enhanced nutrient cycling may have contributed to the high productivity of Carboniferous forests. The underground world. Beneath the visible forest floor lay another dimension of the Carboniferous ecosystem, a dark, humid world of roots, fungi, and burrowing arthropods. This underground realm was just as complex as surface environments, with its own specialized organisms and ecological interactions. Understanding this hidden world reveals how completely arthropods had colonized every available habitat during their age of dominance. The soil structure of Carboniferous forests differed significantly from modern soils. The lack of deep-rooting plants meant most organic matter accumulated in upper layers rather than being incorporated deep into the substrate. The absence of earthworms and other modern soil organisms meant mixing and aeration occurred through different mechanisms. Into this unusual soil environment, arthropods evolved numerous adaptations for underground life. Burrowing millipedes were among the most important underground engineers. These organisms created extensive tunnel systems as they moved through soil seeking food and shelter. Unlike surface-dwelling millipedes, burrowing species were more cylindrical, with reduced spines or ornaments that might impede movement through confined spaces. Their burrowing behavior mixed soil layers, aerated compacted areas, and created channels that could be colonized by other organisms. Centipedes also adapted to underground life, though they generally remain predatory rather than becoming detritivores like millipedes. Soil-dwelling centipedes were often smaller than surface species, with more flexible bodies that could navigate through tight soil spaces. They hunted other soil arthropods, including small millipedes, insect larvae, and mites, filling the predator role in soil food webs. The root zone, the area immediately surrounding plant roots, was particularly active biologically. Roots exude organic compounds that feed microorganisms, creating a nutrient-rich environment. Arthropods feeding on these microorganisms concentrated in the root zone, and predatory arthropods followed. This created dense communities in the immediate vicinity of roots, with population densities potentially higher than in surrounding soil. Mycorrhizal fungi, symbiotic fungi that associate with plant roots, were present during the Carboniferous, though possibly less diverse than in modern ecosystems. These fungi extend the effective root system of plants, helping absorb nutrients from soil. Fungus-feeding arthropods consumed fungal hyphae, regulating fungal populations and influencing the mutualistic relationship between fungi and plants. Evidence for underground arthropod communities comes partly from trace fossils, burrows, and trackways preserved in ancient soils. These structures show that burrowing behavior was common and widespread. The size and shape of burrows provide information about the organisms that created them. Complex branching burrow systems suggest permanent underground residency, while simple straight burrows indicate temporary occupation or passage. The atmosphere within soil pores was likely oxygen-rich due to elevated atmospheric oxygen levels, allowing arthropods to breathe efficiently even in confined underground spaces. This would have supported higher metabolic rates and allowed larger body sizes for burrowing species than would be possible in modern soils. The combination of high oxygen and abundant food made the underground environment highly productive. Underground environments offered protection from surface predators and more stable temperature and humidity conditions. While surface conditions might fluctuate dramatically between day and night, or with weather changes, underground environments remained relatively constant. This stability attracted arthropods that required consistent humidity or couldn't tolerate temperature extremes. Reproduction in soil-dwelling arthropods often occurred underground, with eggs laid in protected locations within burrow systems. This protected developing young from predation and desiccation. Parental care in some species may have included guarding eggs or newly hatched young within burrows, though evidence for such behaviour is difficult to find in fossils. The seasonal dynamics of underground communities probably differed from surface patterns. While surface insects might show distinct seasonal peaks in activity tied to temperature or plant phenology, underground communities may have remained active year-round. The stable underground conditions would have allowed continuous activity, though resource availability might still have fluctuated seasonally. Decomposition in underground environments was mediated primarily by arthropods and microorganisms, with fungi playing a lesser role than in modern soils due to the absence of lignin-degrading fungi. Arthropods broke plant material into smaller fragments, increasing surface area for microbial attack. The combined activities of arthropods and microorganisms drove nutrient release from organic matter, making nutrients available for plant uptake. The evolution of flight. Flight represents one of evolution's most impressive innovations, and insects were the first organisms to achieve it. The evolution of insect flight during the Devonian and Carboniferous periods fundamentally transformed terrestrial ecosystem, creating three-dimensional habitat structure and opening new ecological opportunities. Understanding how flight evolved reveals both the power of natural selection and the environmental conditions that made insect aerial dominance possible. The earliest flying insects appeared in the late Devonian, perhaps 400 million years ago, though the fossil record from this period is fragmentary. By the early Carboniferous, flying insects were diverse and abundant, suggesting rapid evolution once the basic flight mechanism had been established. The question of how wings first evolved remains debated, with two main hypotheses offering different explanations. The paranotal lobe hypothesis suggests wings evolved from lateral extensions of the thorax, fixed flaps that initially served for gliding. These lobes might have helped insects control falls from vegetation or escape predators by allowing brief glides. Natural selection would have favoured larger lobes and eventually the ability to move them, gradually transforming fixed gliding surfaces into active wings capable of powered flight. The gill hypothesis proposes wings evolved from gills of aquatic larvae. Early insects likely had aquatic immature stages, and some possessed external gills on the thorax. If these gills became useful for aerial movement after metamorphosis, perhaps helping with mating displays or dispersal, they could have been co-opted and modified into wings. This hypothesis explains why some features of wings resemble modified gills, including their internal structure. Regardless of origin, the development of articulated, muscle-controlled wings was revolutionary. Early wings were probably relatively simple structures with less sophisticated venation than later forms. Flight muscles evolved to move wings through complex three-dimensional paths that generated both lift and thrust. The neural control systems required for coordinating wing beats, stabilizing flight, and making rapid maneuvers represented significant evolutionary innovations. The Carboniferous oxygen-rich atmosphere facilitated the evolution of large flying insects by making flight energetically easier. Higher oxygen concentrations meant flight muscles could work harder without oxygen limitation. This allowed larger wing muscles that could generate more power, enabling larger body sizes. The largest flying insects appeared when oxygen levels peaked, supporting the connection between atmospheric oxygen and maximum insect size. Wing design in Carboniferous insects showed remarkable diversity. Some species had forewings of similar size that beat in coordination. Others evolved modifications where forewings and hindwings became specialized for different functions. Forewings for power, hindwings for control, or vice versa. Some lineages developed the ability to fold wings over the abdomen when at rest, protecting them from damage and allowing access to confined spaces. The evolution of complete metamorphosis, where larvae are completely different from adults, may be linked to flight evolution. Complete metamorphosis allows specialization of life stages for different ecological roles. Larvae can be optimized for feeding and growth, while adults specialize for reproduction and dispersal through flight. This division of labor increases efficiency and may have contributed to the enormous success of insects with complete metamorphosis. Flight provided numerous ecological advantages. It allowed insects to escape terrestrial predators, access food resources in the forest canopy, find mates over large areas, and disperse to new habitats. These advantages were so significant that flight evolved independently in 3 groups: insects, pterosaurs, and eventually birds and bats, showing that it represents an adaptive peak in morphospace. The biomechanics of insect flight are complex and still not fully understood even in modern species. Unlike birds or bats that use wings as airfoils similar to airplane wings, insects employ a variety of aerodynamic tricks, including leading-edge vortices, clap and fling mechanisms, and rotational forces. These mechanisms generate forces that wouldn't be possible with simple fixed-wing aerodynamics, allowing insects to perform hovering, rapid turns and other maneuvers. Evidence for flight behavior in fossil insects comes from wing morphology and venation patterns. Wings adapted for fast, sustained flight have different shapes and vein arrangements than wings for hovering or slow maneuvering. By analyzing fossil wings, paleontologists can infer flight capabilities and behaviors. Meganeura wings, for instance, show features consistent with fast, powerful flight with good maneuverability. Exactly what would be expected for an aerial predator. The success of flight in insects is demonstrated by their diversification. Flying insects radiated into countless ecological niches, becoming herbivores, predators, pollinators, parasites, and scavengers. This diversity, established in the Carboniferous and continuing to expand through subsequent periods, made insects the most diverse group of organisms on Earth, a status they retain today with over 1 million described species. The legacy of the giants. The age of giant insects ended millions of years ago, but its legacy persists in multiple ways. Modern insects, though smaller, retain many features first evolved during the Carboniferous. The basic body plan, flight mechanics, and ecological roles established when insects were giants continue to define insect biology today. Understanding this connection between past and present reveals how evolution builds on ancient foundations while adapting to changing circumstances. The respiratory system of modern insects, the tracheal network that limits their size, is the same system that allowed Carboniferous giants to reach enormous proportions. The difference lies not in the mechanism, but in the atmospheric oxygen concentration supporting it. This demonstrates how a single anatomical system can produce vastly different outcomes depending on environmental context. The tracheal system was never re-engineered into something fundamentally different. Evolution simply modified what already existed. Flight in modern insects uses the same basic principles established hundreds of millions of years ago. The wing venation patterns visible in Meganeura fossils show clear relationships to modern dragonfly wings. The flight muscles, wing articulation, and neural control systems have been refined and modified, but not replaced. Modern dragonflies are smaller than their ancestors, but perhaps more agile, showing that size isn't everything in evolutionary success. The ecological roles insects filled during the Carboniferous are still filled by insects today, just at different scales. Modern insects are decomposers, herbivores, predators, and pollinators, exactly the roles their giant ancestors pioneered. The detailed plant-insect interactions visible in Carboniferous fossils presage the complex co-evolutionary relationships that define modern ecosystems. Modern ecosystems are in many ways elaborations on themes established when insects first dominated terrestrial environments. The fossil record of giant insects provides crucial evidence for understanding how atmospheric oxygen affects animal physiology and evolution. By studying these ancient organisms, scientists can test hypotheses about the relationship between oxygen and body size, using extinct organisms as natural experiments. This research has implications for understanding not just the past, but potentially the future, as human activities alter atmospheric composition. Modern conservation biology can learn from the rise and fall of giant insects. Their dominance was enabled by specific environmental conditions, and when those conditions changed, the giants couldn't persist. This illustrates how even successful organisms adapted to their environment can be vulnerable to rapid change. The mass extinction that ended their reign shows how interconnected environmental factors can create cascading effects that devastate entire ecosystems. The cultural impact of Carboniferous giants extends beyond science. These organisms capture public imagination because they're both familiar, recognizable as insects, and alien in their enormous size. Museum exhibits featuring Carboniferous reconstructions are popular because they show a time when Earth was fundamentally different, yet still recognizable. This engagement helps communicate scientific concepts to broad audiences. The ongoing research into Carboniferous ecosystems continues producing new discoveries. New fossil sites reveal previously unknown giant insects. Advanced imaging techniques allow scientists to examine existing fossils in unprecedented detail. Computer modeling helps reconstruct how these organisms moved, how they flew, and how they interacted. Each discovery adds detail to our understanding of this extraordinary period. The comparison between Carboniferous giants and modern insects also reveals the constraints evolution works within. Despite hundreds of millions of years of evolution and countless environmental changes, insects haven't fundamentally redesigned their body plan. They remain arthropods with exoskeletons, segmented bodies, and tracheal breathing. This conservatism suggests that some body plans once established are difficult to escape because they impose constraints on what variations are viable. Yet within these constraints, insects have achieved remarkable diversity. Modern insects include over 1 million described species with countless forms, behaviors, and ecological specializations. From parasitic wasps, smaller than pinheads, to stag beetles with enormous mandibles. From aquatic insects that walk on water to those that survive in petroleum pools. Insects have explored nearly every possible variation on their basic theme. The giant insects of the Carboniferous represent a peak in one particular dimension, size, but not necessarily in overall evolutionary success. Modern insects may be smaller, but they're more diverse, more specialized, and adapted to a wider range of environments than their giant ancestors. Success in evolution isn't measured solely by size, but by persistence, diversity, and adaptation. By these measures, insects remain phenomenally successful. The story of the giant insects connects to broader questions about life, evolution, and Earth's history. How do organisms respond to environmental change? What determines the limits of body size? How do ecosystems assemble and function? Why do some lineages persist while others go extinct? The Carboniferous giants offer insights into all these questions, serving as a case study in evolution, ecology, and the dynamic nature of life on Earth. As research continues, our understanding of these ancient organisms will deepen. New technologies will allow us to ask and answer questions currently impossible to address. Future discoveries will undoubtedly surprise us, revealing aspects of Carboniferous ecology we haven't imagined. The giants themselves are gone, lost to time and changing environments. But their fossil remains continue teaching us about life's incredible diversity and the deep history of our planet. The age when insects ruled Earth seems impossibly distant, separated from the present by hundreds of millions of years and multiple mass extinctions. Yet those ancient organisms were real, living their lives in forests and swamps under skies thick with oxygen. They fed, reproduced, competed, and evolved just as organisms do today. Their fossilized remains are not just scientific data, but connections to a lost world, windows into a time so different from ours that it challenges imagination. And in that challenge lies the enduring fascination of paleontology, the science of life's deep past, the study of worlds that once were and will never be again.